Hello, everyone. This is biology, and we are working on module 6, and we are on page 4. We are talking about mitosis today. And what is mitosis? Well, we've done cell replication. Excuse me. We've done cell replication, and we have done protein synthesis. And now we are going to do this process called mitosis. When mitosis is where the cell divides in half. And this is really useful because every time you have a place where you need a new cell or you need growth, this is what happens. Some cells never do go through mitosis. They either come from stem cells, which develop into the particular kind of cell, or they just stay around for your lifetime. Some of these cells are heart cells or stomach cells. Most blood cells don't go through mitosis. Nerve cells don't go through mitosis. But in order to repair your body, for the most part, you need to be able to do mitosis. Okay, so what is mitosis? It is the asexual reproduction of a cell. And it is eukaryotic for the most part. If it was prokaryotic, that means bacteria, it would be called a different process. So there's a lot of similarities between them. It is not mitosis. Now the main phases of a cell's life, it's called the cell cycle. Now the cell cycle includes mitosis, but the main phase that a cell lives in is called interphase. Interphase does not include mitosis. Interphase is part of the cell cycle. And most of the cell's time is spent in interphase. Now, interphase in a normal cell, and of course there's a wide variation in this, but in a typical cell, it will be 18 to 22 hours. So it doesn't take long to go through this process of making more cells. Now, in the cell cycle, of course, that includes mitosis, but interphase is, first you have what they call G1, which is where the cell grows and makes proteins and does anything that it needs to do. Okay, and then comes S, which is the period of time where it makes DNA. It duplicates its DNA. Makes a copy is what it does. And then the last phase, which is also a lot of variation, it's called G2. And this is where it does anything else that it needs to do. In order to reproduce. And so that might be like duplicating its organelles, things like that. Things that it needs to do in order to get there. And this is also called the rest phase. And some cells never go beyond this. And like I said before, those are things like your heart cells, your nerve cells. Now, what does the cell look like? Well, it has a nucleus in the middle. We're going to pretend that this is the cell membrane. Has a nucleus in the middle, and the DNA is like it looks like a tangled strands. Okay, and you could draw like mitochondria, any other things that you have out here if you want to. Okay, now we start in with mitosis itself. And a good way to remember mitosis is this little letters PMAT. PMAT can help you remember what the order is. And the first phase is called prophase. Okay, prophase, our keyword for this is it is preparing. So prepare. Now, what happens is the duplicated chromatin that looked like a tangled mess up in interphase is duplicated now. So the duplicated chromatin condense into chromosomes, which is more what we recognize. These are attached at the centromere. You also have the nucleus dissolve. Okay, so this is where the nucleus was. It is going to dissolve, and then you have these chromosomes that have this X shape up here. Okay, so one of them is now called a chromatid. Instead of a chromatin, now it's chromatid. Chromatin is this tangly stuff right here. But a chromatid is one of these duplicated chromosomes. You see each one is a copy, and they're attached at the centromere. So let's put in there, that right there is the centromere. Then you have these little things called the centrioles, and they start to move apart from each other and develop spindles, which is what these little lines are. Okay, so the centrioles move to opposite sides. And you see these little spindles start to develop. Okay, in the next stage, which is our M stage in PMAT, it's called metaphase. And the key word for this is middle. So what happened in this? The chromosomes line up in the center. And the spindles finish forming. Okay, so what does this look like? We've got our two chromatids duplicated. We've got our centromeres over here, and we've got our spindles, and each one comes up and attaches to the centromere. of each pair. So let's see. We've got this, we've got this. Okay, the next phase is called anaphase. And our key word to remember with this is away. Okay, so these chromatids are separated. And pulled to the centromeres. Oops, that's supposed to be centromere on opposite sides. Okay, so you have your centromeres over here, and those spindles kind of give a pull. And half of your chromosomes go either direction. So they're going towards the centromeres. This is sort of pulling them away from each other. So these are going down here, and these are going down here. The next phase is called telophase. And the key word for telophase is to. So PMAT, you have prepare, go in the middle, go away from each other, and now we develop two nuclei. So the spindles disintegrate. New members form, or not new members, new membranes form around the nuclei. And I say daughter nuclei because it simply means that those are the new ones. Those are the ones that were just produced. And your organelles divide. Okay, so you have single chromatid right here. Over here you have single chromatid. And the nucleus forms around them. And then your organelles kind of go on either side. We've got some maybe Golgi bodies. We've got ribosomes dotting in there, all sorts of stuff we can put in there. Okay, but they've sort of separated in the middle. And the last thing that happens is this process called cytokinesis. And you see cyto, so it has something to do with the cytoplasm. The cytoplasm pinches in half and forms the two new cells. Okay, the chromosomes unwind and become chromatin. And now you enter interphase for these two little cells. And they will start with G1 and starting to grow. Now, this whole process of mitosis takes maybe an hour, 2 hours, doesn't take very long. So here, we'll give a couple of nuclei. And there's that tangled chromatin in it. And we'll do a couple, here's my organelles. And we've got some dots around, things like that. Okay, so now you have 2 complete cells. Now at this point, at the end, there's a last check that goes through. And I'll put down here, last check to make sure that it's genetically accurate. And if it's not perfect, it won't be allowed to go into mitosis. No mitosis again. Like that area in it is like turned off. Now, the exception with this is cancer. The cancer does not have that turn off switch. That's a problem. But in general, your cells, when they're acting healthy and they're the way they're supposed to be, if something is a little wrong with them, they are not supposed to continue into mitosis. They may live long enough to do the function they need to do, but they're not going to be allowed to reproduce and cause a problem in normal situations. Okay, let's go on to talk more about chromosomes. Okay, humans have, let's see, chromosomes, and they come in pairs. So in your cell, you not only have every bit of genetic information you need in order to construct everything in your body, you actually have 2 copies of it in each cell, which is just amazing. That's so cool. They come in pairs. Each pair is basically a similar copy to each other. They are not exactly the same, but these pairs are called homologous chromosomes. No, they're called diploid. We'll get to homologous down here. They're called diploid. Diploid just simply means they're in pairs. Now, if you look at this picture down here, this is a picture of the chromosomes in a diploid cell. One diploid cell would have all of these chromosomes in it, and each one of these is what they call a homologous chromosome, which is right here. Homologous chromosomes are similar, which means they have the same genes on them. But they may differ in what those genes say. And you're like, well, what does that mean? Well, you might have like, for example, take these these 2 chromosomes, these homologous pair, okay? Now, okay, over here, you might have the coding for blue eyes. And over here, you might have the coding for brown eyes. You may carry both, okay? Or over here, you might have the coding for dark skin. And over here, you have the coding for lighter skin. So you see they say different things, but they actually both code for skin color. And over here, they both code for eye color. Now, I don't know which chromosomes actually do that. It's actually multiple chromosomes that decide those things. But this is the idea. Humans have 46 chromosomes. Again, the little pink box shows you. The only one that differs is this one. This is called the sex chromosome. And this is for a male. A male will have an X and a Y. Whereas female will have 2 of these X's, of the longer ones. Okay, so why do we have these 2 different copies of genetic information in our bodies? And that's because we get half of them from each parent. Now, certain cells have chromosomes that are not in pairs, and they are called haploid. So we're going to talk about meiosis in a minute. What meiosis does is it separates these homologous pairs and puts one piece in each cell. And that's what a haploid cell is. It takes 1/2 of each pair. So this goes in one haploid cell, this goes in another haploid cell. This goes in one haploid cell, this one goes in the other one. So the haploid cell still has a complete copy of information, but it only has one. It doesn't have two like the diploid does. Diploid cell, oh, this is supposed to say haploid. Okay, haploid cells are usually called gametes. And those are your sex. Oops, I spelled that wrong. Excuse me. Gametes with the TES. And those are the sex cells. The sperm and the egg. And each parent contributes 23 chromosomes. Chromosomes. Okay, meiosis is the process in which a diploid cell, which has pairs of all these chromosomes, becomes a haploid cell. Actually, 4 haploid cells, which at least one of them has to be useful. Okay, so it's a process. Once again, it's not the word process. The process in which one diploid cell divides into at least one useful haploid cell. And the sciencey word for useful is viable. It means living. It means it'll actually do something. Okay, when two gametes combine, they make a zygote. Remember, a zygote is diploid. Now it has pairs of the chromosomes. And then the zygote will start to divide with mitosis. And will eventually become an embryo, which eventually becomes a fetus, which then becomes a baby. Okay, the steps of meiosis are really, really similar to mitosis. The cells go through the same process. The biggest difference is in meiosis 1, which is there's 2 stages. There's meiosis 1 and meiosis 2. In meiosis 1, you're going to be separating pairs of chromosomes. You're not pulling apart the chromatids like you did in mitosis. You're separating the pairs together so that one homologous pair goes into each of the 2 cells. In meiosis 2, then the 2 chromatids are going to be separated. So that's what happens. So in meiosis, the first stage, you still have prophase 1, metaphase 1, anaphase 1, and telophase 1. So it's PMAT still works, but different things happen. So what happens first in prophase 1 is that the homologous chromosomes line up together. And they call what you know is a tetrad. Now remember, these are now duplicated. So you have these 2 things together and they're attached in the middle by a centromere, just like with mitosis. But the homologous chromosomes line up together and you can see it down here. See, you've got these 2 chromosomes that are lined up, but they're usually closer than that. And then what happens is that pieces, and you can see it down here, pieces of one homologous chromosome will trade with another homologous chromosome. So for example, if this gene right here said brown eyes, and this one right here said blue eyes, and then they cross over and they trade, now over here, I think I said brown first, this one now has the brown eyes and this one has the blue eyes. And this ensures genetic diversity. So this makes sure, we're going to put here, ensures genetic diversity or differences. And this is why children are never really quite the same as their parents. They might be a combination or they might be crazy, that they might look really different, not just the same as their parents. So what is crossing over is that segments exchange between chromatids. And this makes new combinations. And sometimes those new combinations look very different from the old combinations because genes are incredibly complex. Okay, in metaphase, you've got these two pairs. Remember, picture your tetrads down here. In metaphase, the tetrads line up in the middle. Anaphase, which is away, say each pair, one pair of each goes to each side. And in telophase and also cytokinesis, then you have two new cells are formed, just like in mitosis. The nuclear membranes reform, and then cytokinesis pinches them off. So now you have two daughter cells, each with one set of homologous chromosomes, not two sets like normal, but one set of homologous chromosomes, but each of them are duplicated. Like each of them are duplicate. So steps of meiosis, second stage. Okay, so prophase, the spindles form. Remember, they do not have to duplicate. I think something happened to no. First, you have the spindles form in each daughter cell. Something's going on with this. Let me change it down a little bit. In each daughter cell, in metaphase, they line up in the middle. So the pairs, I guess they are pairs line up in the middle. In anaphase, then those pairs are pulled apart. And a single chromatid goes to each side. And then in telophase 2, again, there's cytokinesis in here. Now you have both daughters pin the cell. Let's see. Well, you have two daughter cells each. And that means altogether you have 4 cells. Each has one little chromatid in it. Now, what happens here between men and women is very different. The male, all 4 sperm becomes a sperm. All 4 cells are viable. That means they're useful. 4 viable cells. Okay. They develop a flagella so that they can move. Okay, the female, you have only one of those 4 cells actually is useful. Okay, you usually have one of them, one of the 4 cells is bigger and stronger, got more of the strength. The other 3 are known as polar bodies. Polar, polar bodies, and they disintegrate. So you just have one really big strong cell, and the other ones just kind of fade away and disintegrate. So let's look at the last page. There's not a lot to copy. Whoops, let me get rid of these. Select all. Okay, I just need to get rid of the. Okay, let's look at the chart on the last page. These kind of go through meiosis and mitosis because I wanted you to be able to see it. And there's lots of good videos. The biggest difference between meiosis 1 and meiosis 2 is right here. You see this? That here you have pairs separating. And then down here, you have the chromatids separating. That means that the pairs break apart. Down a little bit more. Pairs break apart. That's the biggest thing to remember, the difference between meiosis 1 and meiosis 2. Meiosis 1, you've got those pairs separating. In meiosis 2, you have the actual chromatids separating. So here you have 2 cells, 2 daughters. Here you have 4 daughters, because each one of the daughter cells from up here divided in half to make 2 of these down here. Okay, that finishes up the notes for module 6. Thank you for watching, and if you have any questions, please let me know.